Welcome to Ceremonial Hall of University of Sofia St. Kliment Ohridski, the oldest Bulgarian university. I am Stoyan Shishkov, Decan of Faculty of Biology and co-organizer of the Congress. Dear Vice Rector, Sir David, Professor Vitanov, Professor Zhelev, in these few days of the World BioDiscovery Congress, scientists, inventors, representatives of bio companies, university lecturers, and students will have opportunity to share our achievements and experience, as well as our opinions in various fields of the life science and medicine. I'm sure our meeting will be interesting and helpful for us as well as for the society, and new ideas are about the emerging. On the last day, I'm delighted to welcome you in my university and our faculty. We will have the opportunity to see the faculty, and we will have the final session of the Congress. Now I would like to invite Professor Zhelev from University of Aberdeen, Dundee, to tell you more about the Congress. Professor Zhelev.
Vice Rector, Sir David, Professor Shishkov, Dean of Faculty of Biology, dear friends, дорогие друзья из Беларуси и России. I feel a little bit emotional in this ceremonial room because, 30 years ago here I received my diploma from then the rector of the Sofia University, but I'll try to keep my emotions. I'd like to welcome you all. We call ourselves a Congress. A Congress is a big name. And as you can see, we are around 80 specially selected people here. We are running this event for a few years now, maybe 5 years. We've done it in Baltimore. Last year was in Dundee, and this year is in Sofia. The reason we have chosen that format is to have less people than other congresses, because we find it more efficient. So I hope we'll enjoy the event. It will result in new ideas, new grant applications, and new friendships. So with that, I'd like now to invite the host, the Deputy Rector of the University, Professor Vitanov, who is, as all of you know him very well, he's Deputy Rector for Research, to welcome the guests and officially open the Congress. Professor Vitanov. Yes.
Dear participants in the World BioDiscovery Congress 2017, it's indeed a great pleasure for me and great opportunity to welcome you here in Sofia and in our alma mater, Sofia University St. Kliment Ohridski, on behalf of the president of our university, Professor Anastas Gergikov. I would like especially to congratulate and to thank the chairs of this event, Professor Shishkov, Professor Ugrinova, and Professor Zhelev, and the International Organizing Committee for organizing this very timely and very interesting event. With about 100 participants from over 10 countries from around the world, this event is about to become a major scientific event in this field, not only for our university and for our city and for our region, but also for Europe, I think. There are many scientific events held every year at Sofia University, and especially in this nice aula. But something that really makes this event special is the involvement of both academia and industry. It's indeed very nice to see so many distinguished speakers from both academia and industry. As the motto of the Congress says, from research to discovery, it's very important nowadays to discover ways to implement scientific excellence in practice to the commercial stage. I'm looking forward to listen to the biopharmaceutical industry to share their experience in developing a business on the basis of university research from the lab to the commercial stage. It's also quite important for me to see that there are so many young scientists here from Sofia University and the Bulgarian Academy of Sciences who have the opportunity to discuss their results with world experts in their fields and get advice on their future scientific and possibly business development. I'm looking forward also to share experience with leading experts from these 10 countries, from universities around the world, about how to involve businesses in university education, because this is also very important for our university. I appreciate really the size of this event, because I've been myself to many conferences of huge size and small size, and now know how important it is that the opportunity that everybody can speak to everybody. So in conclusion, I would like to wish you a very interesting and fruitful conference and a very pleasant stay in Sofia. Thank you.
I'd like to introduce the first speaker for today, the guest of honor. He's well known around the world, and I think he's also very well known in Bulgaria. So, Professor Sir David Lane has been in Bulgaria 10 years ago, or a little bit more than that. Then we had a very fruitful discussions with young scientists, which are now, some of them are rectors, head of departments. He visited Bulgarian Academy of Sciences, Medical University of Sofia, and the Sofia University, and had a very fruitful discussions with a lot of scientists. As a result of that, we arranged some reagents to be transferred to these universities, and a lot of useful publications have been generated, outputs, books edited. So he has been involved in Bulgarian science for many years. And also, I don't know, maybe he made a mistake, but he has employed a lot of Bulgarians in his lab. And I'm one of the people who benefited when I joined his lab some 25 years ago. So you learn a lot. You can learn a lot from him. You probably know that in terms of science metrics, he is one of the top scientists in the world. You know, very popular now in Bulgaria, not to look at the person, but just to measure him in terms of citations and H indexes. And in that respect, he is astronomical citations and H indexes. But not only that, not only he's a good scientist, he's been very much involved with the real life. He's been involved with the biopharmaceutical industry, a lot of collaborations. All this time, seeing his research being translated into practical applications, into medicines. He himself founded several companies in Europe and also in Asia. And the remarkable thing is, this company still exists. You might laugh, but you know, the life out there is not secure for the biotech companies. They are not like universities. In different business climates, companies go up and down. A lot of biotech companies disappeared. And all companies which Sir David has founded still exist, which is a big achievement. I have a big list of awards he has received. He's an honorary fellow, professor, in many academies. I'll just mention some more important, like Bulgarian National Academy of Medicine, for example. And a lot of awards, including the so-called Small Nobel Prize. So it's a person we can learn a lot from, and he's here at our disposal for 3 days. It's very difficult to catch him. At other congresses, like the one in Chicago, people were struggling to talk to him. Even I wasn't able to talk to him because of this circle of people around. Here we have the chance, after this lecture, during the coffee breaks, to ask him questions. Please go forward, because there's a lot to learn. Sometimes one question will save you reading hundreds and thousands of papers, because there's a lot of good papers there in the library. The problem is you can't ask them questions. They only give you some answers, and they don't reply to questions. So use this time to ask him for advice. He's going to look at our posters and evaluate them. So it's a big privilege to have Sir David at our conference, and for us to have the chance to talk to him. Because with that, I would like now to invite Sir David to give his keynote speech. Please, Sir.
Well, thank you very much, and it's really a pleasure to be here. Thank you very much, everybody, for inviting me and for the very nice opening remarks. I met Nicola and Daniela many years ago. I won't say quite how many to embarrass us, but we all kept very young looking, so that's good. And they were my two first scientific employees of the biotechnology company I set up in Dundee, Cyclocell, which is still going, as Nicola said. And actually we'll hear a wonderful talk later on from Daniela about the progress they're making in bringing molecules all the way from basic science to clinical trial. And it's certainly one of my ambitions as a scientist to see the work that we do translate into being useful. It's a hard and long journey, but one of intense interest, and I'll talk about it a bit today. I first discovered p53 as a complex with sp42 antigen in 1978, and our paper appeared in 1979, so I've been working on the system I'm going to talk about for most of my life. It's older than my children, which is always a bit sobering. So why are we so fascinated by p53? It's really because it seems to be the principal gene that protects us against cancer. So people who inherit a mutation in the p53 gene have a huge increased incidence of developing cancer. It's enormous influence on their susceptibility to many different sorts of tumors, and that syndrome is called the Li-Fraumeni syndrome. And as we've got much better at sequencing DNA, we realized that mutation in p53 is more frequent even than we thought in the early days. And even 10% of children who have cancer turn out to have mutations in p53, not necessarily inherited, but sometimes new mutations as well. So what does this tumor suppressor protein do that is so effective in protecting us people from getting cancer? Well, it's a very, very responsive transcription factor. Its activity is stimulated by DNA damage, by radiation from ultraviolet light, by radiation from ionizing sources, but also by a whole variety of different stimuli, in particular the activation of oncogenes. So it acts particularly to recognize aberrant proliferation in cells and to switch it off. And it does so by accumulating in the damaged cell and then moving onto the DNA at the specific binding sites for p53, and then activating a very complex transcriptional program that regulates apoptosis, cell cycle arrest, senescence, DNA repair, metabolism, feedback regulation, etc. And in the conference we just held in Singapore, which had 350 delegates from throughout the world, people begin to really drill down into the p53 program, and there's a kind of essential program of at least 100 genes that are activated directly by p53 across cell types. If you look at different cells in the body, though, some turn on different programs than others. So, for example, a thymocyte in which p53 is activated will die by apoptosis, but an epithelial cell will arrest and stop growing, but can then recover. So we see different responses in different cells and tissues. And really the components of this signaling pathway are twofold. One is the blocking of degradation of p53 by the ubiquitin E3 ligases, MDM2 and MDM4, and that process is itself subject to very complex post-translational modification of both MDM2 and p53, and then the activation of p53 as a transcription factor, again involving post-translational modifications. So in tumor cells, this is the kind of signaling pathway we see, and this is what's creating the pressure for mutation, because if you mutate p53, you lose all of these responses, and the cells are therefore able to proliferate in the presence of an activating oncogene or a stressful environment. But in the normal cells of our body, in fact, the pathway is much simpler. We're essentially making p53 in an almost futile manner and just degrading it, so that we see in normal cells of the body actually minute amounts of p53. If you do a proteomic analysis, it doesn't exist. So you have this incredibly important protein, and yet you can't find it except in tumor cells where it's overexpressed. And the interest is to understand the regulation of this pathway and then to try and translate that into therapy. So we essentially have a huge discovery that half of all human tumors are missing this pathway. And how can we turn this into a therapy is a challenge that we all face in the community.
става въпрос за това, че един наш колега, който в момента е професор в Абърти университет в Дънди, Шотландия, има контакти с нас от дълго време, още повече, че той е българин, завършил е Софийски университет, молекулярна биология и от доста време живее и работи в Обединеното кралство. Покрай него има и още доста българи, които отидаха там и се занимават с наука, с биология. Той по-специално се занимава в областта на противотуморните лекарства и изследвания. Има собствена компания за производство на такива лекарства и прояви интерес. От няколко години връзката ни се заздравява, присъствие на наша конференция международна младежка и оттам дойде идеята да се направи един такъв конгрес в България, което има за цел, освен да популяризира научните изследвания в Софийския университет и в някои други научни институции в България, така и да се създадат връзки с колеги от чужбина, които до сега не сме имали контакти и евентуално, тъй като тук присъстват представители на различни фармацевтични и биологични фирми от световен мащаб, да се направят контакти с наши учени, т.е. бизнеса, международния, да може да се заинтересува от нашите изследвания и евентуално да се създаде една колаборация, да се направи колаборация за финансиране на научните изследвания в България, респективно в Софийския университет. Значи конгресът поставя на преден план основно връзката между научната общност, между изследователските екипи от света с съответните фирми, които са заинтересовани от новите открития, които биха довели до развитие на нови лекарствени средства и естествено по-нататък фармацевтичните компании пък имат интерес да са първи и да се развиват и те на базата на нови изследвания и на нови продукти в областта на медицината.
Кои са основните предизвикателства пред биологическите науки, пред биологията в началото на 21 век?
Основното предизвикателство може би е това, че тя е изключително бързо развиваща се наука в всички области. С този инструментариум, който в момента разполага, с всички възможности на молекулярната биология и на биотехнологиите, могат да бъдат разрешавани редица въпроси, като борбата с туморните заболявания, генната терапия, която все повече и повече напредва и има приложимост, макар и изследванията да не са финиширали, така да се каже. Освен това, съвременният свят дава възможност за развитието на биотехнологиите, на индустриалните биотехнологии, на растителните биотехнологии с цел подобряване на живота на обществото.